All right, we are finishing up chapter 2 here. We're going to continue through our list of characteristics of cancer cells. And I wanted to start the second half here talking about loss of cell cycle regulation, which you find in pretty much all cancers. So we've already talked about some of the things that will allow cancer cells to keep dividing, but here we're really focused on, what is it that tells a normal cell to divide in a typical situation. So you may have seen some of this before, but I just wanted to review some of these cell cycle control mechanisms. And so normally things like growth factors are going to be what stimulate cells to divide. So of course there's all kinds of different growth factors. For example, epidermal growth factor. So this would be, secreted by certain cells in your body. Would bind to receptors on the surface of other cells, initiate a signal transduction pathway that is going to kick on transcription and translation of genes and proteins that would then be required for cell division. And what we often see in cancer cells is this process is broken. It's kind of locked in the on position. So for example, in this particular example, the receptor protein itself has become mutated so that it no longer needs to receive that growth factor signal. It's just constantly going to signal downstream, telling the cell to divide, even in the absence of that external stimulus. So things like that are very, very commonly seen in cancer cells. And we'll get into some of these pathways in a little bit more detail later and come back and revisit this idea. But in terms of cell division, just to remind you the basics of the cell cycle, right? You've got your mitosis, your cell division, and then interphase, which consists of G1, S, and G2, S being synthesis phase, right, where DNA replication is occurring. And I wanted to illustrate here, you see this red line, this is a example of a checkpoint. This particular checkpoint is referred to as the restriction point, as kind of arguably maybe the one of the most important cell cycle checkpoints. But you can think of this and other checkpoints. There's actually 4 or 5 throughout the cell cycle as kind of red lights where the cell is going to have to stop as it's progressing through the cell cycle, and it will only be able to proceed past that checkpoint if the appropriate signal is received telling the cell, yeah, it's okay to continue. And sometimes that signal may be as a result of some internal bookkeeping, right? We want to make sure we have the right number of chromosomes, or make sure the DNA is not irreversibly mutated, or there's lots of different possibilities there. But at this restriction point, it's actually possible for the cell to exit the cell cycle and enter what's called the G0 non-dividing state. So most of the cells in your adult body are going to be in this G0 state. They're not actively dividing. Cells in your adult body are only going to divide to replace lost or damaged cells usually. And so, what is it that controls the progression through the cell cycle and whether or not cells are going to be able to move past these checkpoints? Or is this class of proteins called CDKs, or cyclin-dependent kinases? And just to remind you, kinases are a broad class of enzyme that like to phosphorylate other molecules, other proteins, for example. And they'll do that by taking a phosphate group from ATP and then attaching it to the target molecule. And so we have these cyclin-dependent kinases that, as that name implies, they're only able to work as active kinases when they're bound to their partner protein called cyclin. So you have CDK plus cyclin together. And also just wanted to point out that on the flip side of this coin, we have enzymes called phosphatases that can remove phosphates or dephosphorylate. So you have kinases adding phosphates, phosphatases removing them. But we're going to focus here on a kinase, on the cyclin-dependent kinase in particular. So, one way cyclin-dependent kinases would work, for example, at that restriction point that we were just looking at, is by phosphorylating a specific protein. In this case, the protein is called RB, retinoblastoma protein. And so you can think of retinoblastoma protein as the red light, right? It is basically preventing the cell from going into S phase of the cell cycle. We'll talk later on about how it's actively doing that. But if the cell has received a growth factor saying, yeah, you do need to divide, right? You need to go through the cell cycle, go through S phase, get ready for a mitosis. If we have this growth factor showing up, binding to a receptor on the cell surface, telling the cell to do that, this signal transduction pathway will lead to the activation and production of cyclin CDK, which will then phosphorylate that retinoblastoma protein, right? As we just talked about, taking a phosphate from ATP, or multiple phosphates, adding it to retinoblastoma, and then that is going to inactivate that retinoblastoma protein, effectively turning the red light green. So you'll see some variation on that at any of these multiple cell cycle checkpoints. Cyclin CDK showing up, phosphorylating some protein, some enzyme, and then that's going to change the biological activity of the cell. And so I mentioned there's multiple checkpoints, so just to show you some others, you have several DNA damage checkpoints, right? One basically right after the restriction point, one in the middle of S phase, one right before we go into mitosis, which as the name implies, right? We're checking for DNA damage, making sure that the DNA is not mutated beyond repair. This one is also considered a DNA replication checkpoint, so make sure that we actually replicated all the DNA before we go into a division. Right in the middle of mitosis, you have a spindle checkpoint where the cell is making sure that the mitotic spindle is attached properly to all the chromosomes. So again, we have these red lights, these quality control measures in place, and the cell can only move past these checkpoints if you have active CDK cyclin there to phosphorylate something and allow the cell to proceed. Oh yeah, I forgot even another one here at the called the G2M transition checkpoint. So anyway, you don't have to memorize the location or the names of all these checkpoints, but I just want you to be aware that there are quite a few of them. And so the basic idea here is in cells typically you'll find CDK part of the cyclin CDK combo almost always present in most cells. So it's always there, but the cyclin portion will kind of come and go. It'll be degraded once the cells move past a checkpoint, and then we would have to produce new cyclin if we wanted to go through another round of cell division and get past that checkpoint again. And I should also point out there's lots of different flavors of cyclin in CDK, so it's not just there's one cyclin, one CDK. So different cyclins will pair with different CDKs to get past some of these different checkpoints. And I wanted to zoom in on a little bit more on what detail of what's going on here with cyclin CDK activation, because it's not quite as simple as, oh, they just have to both be present and paired with each other. There's actually some phosphorylation, dephosphorylations that have to happen for it to become fully activated. So you do have to have both proteins, the CDK and the cyclin. They do have to bind to each other. But then a kinase comes in and adds a couple of phosphates that are actually considered inhibiting phosphates, shown in white here. So they will actually prevent the CDK cyclin from having any biological activity. Then an activating phosphate will be added. So you have one activating, two deactivating, and now for this complex to actually become active as a kinase, we have to have a phosphatase enzyme come in and remove the two inactivating phosphates. So once those are gone, now you have an active cyclin CDK complex that can actually start adding phosphates to other molecules. So kind of an interesting little dance going on there where we add some phosphates, remove some phosphates to get full activation. You might ask, well, why is it doing that? And I think it's just giving the cell a little bit extra, another extra level of control where it can it can have that cyclin CDK complex produced and ready to go, but not fully active. And then all we have to do is come in and snip off those 2 phosphates, and it can be, ready basically then at a moment's notice. So the fact that cancer cells have lost control over many of these restriction points, so typically what we see is cancer cells are not stopping at these checkpoints. And maybe for numerous reasons, as we discussed, right? Maybe the receptor is hyperactive and constantly sending the signal, constantly activating cyclin CDK, or maybe the cyclin CDK proteins themselves are hyperactive. But at any rate, cancer cells are usually not stopping at those checkpoints. But that phenomenon can be used in certain cases to actually try to get rid of cancer cells. So, for example, there's this combo drug treatment using these compounds called staurosporine and camptothecin that can be used to try to chemically distinguish between normal cells and cancer cells and just kill the cancer cells. So what staurosporine does is it will typically block normal cells from being able to go into S phase. And then camptothecin will kill cells that do enter S phase. So what happens if you treat normal cells, right? The staurosporine should stop them before they enter S phase, so the camptothecin doesn't really have any effect on them. And we wash out both drugs and those cells are fine. They can resume the cell cycle. Whereas cancer cells, because they're not stopping at that checkpoint, even in response to staurosporine, they'll cruise right into S phase regardless. Then the camptothecin will kill them. And so we get this nice preferential destruction of cancer cells in this particular instance. So kind of using that loss of cell cycle control to our medical advantage. Now, the 6th characteristic is genetic instability, which, as I said, is just accumulation of mutations and chromosomal abnormalities. And as we also talked about, right, normally mutations or chromosomal abnormalities will trigger apoptosis, but because cancer cells have often shut off the apoptosis pathway ironically through mutation, they're able to stay alive. And I just want to remind you, I'm sure you're aware of this, but we'll talk a fair amount this semester about mutation in cancer cells, right? Just remind you, why are mutations in the DNA a problem? Well, keep in mind, right, it's your genes in your DNA that are coding for specific proteins. So if we make changes up here in the DNA, those changes end up in the RNA, in the messenger RNA, and then can end up in the protein. So basically, if we mutate the DNA, we're either changing or potentially inactivating specific proteins in the cell. So that's really one of those hallmarks of cancers, is we have typically mutated multiple genes, changed multiple proteins. And if you think of this kind of historically, it's kind of interesting. When people first started becoming aware of this whole idea of mutations and cancer, it was a bit of a chicken and the egg type of problem, because researchers didn't know, does a cell become cancerous because the DNA in that cell has changed? So they knew the DNA was different in cancer cells than in normal cells. But were those changes causing the cells to become cancerous, or was it the fact that the cells had become cancerous now was causing damage to their DNA? So it seems obvious to us now, but somebody had to sort that out. And basically what they did was they took DNA from human cancer cells, chopped it up into little pieces, and then injected that cancer cell DNA into healthy, normal fibroblast cells that were growing in tissue culture. And what they would find was, after introduction of the cancerous DNA, that was basically transforming, causing some of those normal healthy cells to become cancerous, which would argue, it's the DNA, this cancerous DNA is causing the normal cells to become cancerous cells. And they found, they can even take those cells from the petri dish, right? Inject those into mice, and yeah, you're actually getting a tumor there in vivo as well. So that kind of solved this problem of, yeah, it's the DNA, changes in the DNA is what's causing the cancer. Sometimes you'll see this referred to as transfection, so a normal cell being forced to become cancerous by introducing this altered DNA. And of course, there's lots of different types of DNA damage that can occur, right? Could have, single point mutations. You could have depurination or deamination. Depurination is where you're actually removing a purine base. You have a base missing. Deamination, you're removing an amino group. Can do interesting things like convert cytosine into uracil. Of course, you don't usually find uracil in DNA. It's usually only found in RNA. UV, the energy in UV radiation, we'll talk about later, can cause the formation of these bonds between adjacent pyrimidine nitrogenous bases, or dimers they're often called, that can then distort the DNA and cause problems during DNA replication. So anyway, there's lots of different types of DNA damage, but we do have mechanisms in our normal healthy cells to try to repair most of these types of damage. So for example, we have excision repair pathways, it's called. So in the basic gist of excision repair is you have what's called an endonuclease comes in, finds the damaged DNA, let's say a pyrimidine dimer. It chops out that damage, so you're left with a gap, and then DNA polymerase can simply fill in the gap using the complementary DNA strand as a template. Ligase comes in and seals it up, and boom, we're back to normal. So you have these repair pathways kicking away in the background. But what we see in a lot of cancers, and again we'll get into this in some later chapters, is some of these repair pathways themselves are defective or have become mutated. So these kind of damage can no longer be fixed in cancer cells. Sort of a subtler version, DNA mismatches, which are technically not considered damage but are a problem. So, if we imagine we have a normal AT pair in our DNA, let's say due to a replication error, we paired a cytosine with adenine instead of a thymine, so now we have this mismatch. Well, if we're able to recognize and repair that before we go through another round of replication, then everything is fine. But if we don't, and we do go through another second round of DNA replication before that mismatch is corrected, now we're basically going to cement that change, because we're going to pair the cytosine with a guanine. So we're going to go from an AT pair to a GC pair. So now we've got basically a permanent mutation in that one nucleotide in the DNA. And again, there are these mechanisms for repairing mismatches that often become damaged or ineffective in cancer cells. So, the defects in these normal repair and identification processes are what are allowing this genetic instability to occur, these cancer cells to accumulate lots of mutations over time. And so in addition to these kind of subtler mutations, we often also see bigger chromosomal abnormalities, weird numbers of chromosomes, broken chromosomes, weird translocations. So here you can see a karyotype from a cancer cell, and you can see there's all kinds of weird stuff going on. So we've got like 3 copies of chromosome 8 and 1 copy of chromosome 13, and weird translocations that have happened. We basically got like 1 copy of chromosome 13 or 18 is basically attached to chromosome 1, or maybe that's chromosome 5. Anyway, right? Things are not as they should be. So some of these larger scale problems are believed to come from, for example, spindle checkpoint errors. So this would be the checkpoint, as I mentioned earlier, where you're making sure the mitotic spindle is correctly attached to all the chromosomes, right? It's the spindle that's going to help make sure segregation happens correctly. So if that checkpoint is not working properly, you're going to get lots of non-disjunction of chromosomes and end up with lots of aneuploid cells, basically. And in terms of some of these like broken, damaged, weirdly translocated chromosomes, you see this, for example, in Philadelphia chromosome, where you've got um, portions of chromosome 9 and 22 that have translocated with each other, and that leads typically to specific types of leukemia. Some of these weird translocations are commonly caused in virus-infected cells, where it seems like the virus is triggering cell division at such a rapid rate that for whatever reason you're getting these unusual and unfortunate translocations. All right, we're getting there. Last two, deregulation, dysregulation of cellular energetics. And this was referring to this relatively new and not entirely understood finding that a lot of cancer cells actually seem to favor glycolysis over full oxidative phosphorylation. And we don't entirely know why that's the case. There's some thinking that some of the intermediates produced during glycolysis can be diverted into some of these other biosynthetic pathways that are producing macromolecules that are in short supply in these rapidly dividing cancer cells. And we do find something similar going on in many populations of rapidly dividing embryonic cells as well. So it seems like this may be a pretty typical phenomenon found in rapidly dividing cells, either healthy or cancerous. Could also be tied to the fact that some tumor environments are found to be relatively hypoxic, meaning lacking in oxygen, and this may be caused by just weird vasculature, disorganized vasculature around the tumor. And so, of course, if you're lacking oxygen, oxidative phosphorylation is not going to be an option, whereas glycolysis will still function properly. And then lastly, I just want to touch briefly on evasion of the immune system. So normally, there are immune cells that can hopefully recognize and destroy some cancers, and there's quite a few different tricks that tumors can use to escape recognition by the immune system. So they may just have low immunogenicity, meaning there's not anything unusual for the immune cells to recognize. And this kind of ties in with the second one here too, which is, because cancer cells are self cells, that makes it more difficult for the immune system to find something unique to attack, right? Because we don't want our immune cells to attack our normal healthy cells. So there has to be something recognizably different about the cancer cells, some specific antigen that they can target. Even if there is an antigen, of course, tumor cells may be able to mutate those antigens so that the immune system no longer recognizes them. There are cancer cells that actually produce specific proteins, molecules that can suppress immune cells directly, or some tumors can secrete actual physical barriers that will prevent the immune cells from gaining access to them. So extracellular matrix, for example. So anyway, we'll bump back into some of these ideas again in later chapters. But I think now you've got a pretty comprehensive view of, what's going on with cancer cells, what are some of the key changes that happen, and how do some of these things occur.